Thank you so much for the kind introduction, Dirk. And thank you for inviting me. I'm very honored to talk to your world-known center. And it's kind of weird these days to talk to an audience you can't actually see, but I hope it goes well. And so today I want to talk to you about changes in language network and primary progressive aphasia. And, you know, a lot of our understanding of language network and aphasia have come from stroke-related aphasias. And primary progressive aphasia as a neurodegenerative disease has really opened a different horizon for us to look at language function, because the pathology really affects the brain differently. So we've learned a lot. And then as a disease, we are trying to find out what's wrong in these individuals and how we can help them. So I will start by my disclosure slide. I do have a lot of conflicts, but none of them are of interest. And these are the list of grants that have supported our studies. So to give you an outline, I'm going to talk a little bit about language network localization and its challenges. We are still trying to understand the boundaries of language network. It's an ongoing task, but we know much, much more than, say, half a century ago. And then I'll talk to you a little bit about functional imaging. That's mostly what I'm focused on talking about today. And I know we have a wide audience. We have world-class aphasiologists, and we have neuroimagers, we have linguists. So I'm going to talk a little bit about language assessment for those of you who don't deal with it on a daily basis. And then a little bit about primary progressive aphasia, what happens in network connectivity in PPA, and some findings within the right hemisphere, because everything in right-handers, as you know, happen in the left hemisphere, and some thoughts about implications for therapeutic interventions. So in 19th century, things were much easier. We learned from Paul Broca that the language expression or production is in the ventral or lower frontal lobe, what was called later Broca's area, and then later Wernicke described the patients with stroke within the superior temporal gyrus who had trouble with understanding language. So everything was like expression frontal and comprehension posterior. But then we learned from Lichtheim that these two areas are connected to each other with arcuate fasciculus, as you know. And then fast forward, we get to the Gershwin model and the Wernicke-Lichtheim-Gershwin model, which shows how language is perceived or understood and produced. And as you see with Gershwin's model, other areas were added to the mix. You know, you have the temporal junction, you have the auditory cortex, visual cortex, and motor cortices as well. What was missed for a long time, however, was because the models were based on stroke, not much was focused on what's happening within the white matter, because stroke follows the vasculature of the brain, not the networks. So what happens is when you have a stroke, you not only have damage to the cortex, but you have damage to white matter as well. So that means if you have a damage to white matter, areas that are connected to that white matter track, and then can be much remote, they can be affected as well. So you may get a lesion in the back part of the brain and then have agrammatism because there's no connection with the front of the brain where grammar is processed. And this was a beautiful study that was done by Nina Drunkers and others. They actually scanned the brain for Moussioton, the Le Brunier brain, which is still available. And as you see, so left is left. You see in the axial cuts that
there is a lot of white matter damage on the left side, going through the superior longitudinal fasciculus, perisylvian area. So the fact that this patient had certain symptoms, it wasn't because of just damage to the Broca's area, which we see on the surface, but there was a huge disconnection with the posterior areas of the brain as well, which tells us there is more than just one localization, but the whole network that's involved. And then throughout the past 20, 30 years, we've learned a lot about, details of structural changes in the brain in stroke and other diseases. This is a slide from Stephen Wilson's paper where, when we started to learn that there are all these tracks, the superior longitudinal fasciculus and arcuate, you have the external capsule, you have the oncinate, and based on Greg Hickock's and David Popel's theories, now we know that the language is not only organized anterior, posterior, but also dorsal and ventral, with dorsal being more related to phonological and syntactic processing, and the ventral for lexical semantics. And so this model still works, but still it may miss some details, and I'm not going to go through all the details of models of language network. But this, for our purposes, works. So there are a few more that I'll show you. So aside from the superior temporal gyrus, the role of middle temporal gyrus has been shown to be very important, both in production syntax and in comprehension and lexical semantics. And then you see the two pathways for repetition and sentence production. This is based on Federici and Gerhan's review. And then we have a beautiful paper that came out of your center 2 years ago, a year and a half ago, where we had best of two worlds. So both the cortical changes and white matter changes were looked at. And you see this will elucidate actually a lot of more details. For example, in sentence comprehension, you see tracks that are more dorsally oriented, although you do have loss of cortex in the temporal lobe. And then word recognition, you see ventral white matter involved, although both have temporal lobe damage as well. So this confirmed a lot of the dorsal and ventral organization of language. In addition, you see for each of these language measures, you can see there are different anatomical correlates. So in terms of fMRI, and I'm sorry for those of you who know a lot about fMRI, this is for people who may not know the details. So functional MRI is based on changes in oxygenated hemoglobin. So when a person is doing a task in the scanner, there's increased blood flow to that area. That means there's increased oxygenated hemoglobin to that area, which changes the magnetic field in that area. And then based on that, we can look at the changes in the hemodynamic function. And that'll show up like this curve, and then when we average it and clean it up, then we can see the activation areas in the brain. This would show a task that we did years ago for naming, where when you show a person a picture, you have the hemodynamic response going up, and then it's the same thing if we repeat the task and then we average it, we can see the areas of the brain that are involved in, for example, a task like naming. This is called task-based functional MRI. And we did a lot of research with Dirk in 2000s, looking at neurocorrelates of verb and syntax processing, the dorsal pathway, which at that time we didn't quite know what the details were. For example, we thought verbs that are very crucial for sentence production are processed in the frontal lobe, but then all we were getting was in the temporoparietal junction. For more difficult verbs like the mother is reading a book to the daughter, has 3 components, you see more activation within the temporoparietal junction. And then later, the research in Cindy Thompson's lab showed that, this is how it works in terms of having the verb retrieval argument structures and putting the phrases together and the words together. And this kind of explains how the connectivity is for sentence production. And so for resting state, functional connectivity, in aphasic patients, as you know, it's difficult to train them. They may have trouble scanner to do the task. So resting state was pretty impressive and got a lot of popularity in 2000, later in 2000s. So I got more interested in it, and then there are different ways to look at connectivity at rest. You can look at it based on region-based analysis, so ROI-wise, pair-wise analysis. You look at the hemodynamic change fluctuations in blue here in this area, and you look at the hemodynamic fluctuations in the red area, and then you look at the correlation between the two. The more correlated they are, the more connected they are supposed to be. And now, so this is called the node-based pairwise analysis. There's another way to look at hemodynamic fluctuations, and that's to get one RI or one seat and ask the question of what areas, what voxels in the rest of the brain are in sync with this area. Then that'll show you every voxel in the brain that is in connection with this certain seat. So this is called the seat-based or voxel-wise analysis, a whole brain analysis. These are hypothesis-based because we are picking the actual region of interest. There are data-driven ways to look at the resting state information, and this is independent component analysis is one. And that would be if you imagine that the brain, this is what, for example, what we record from an EEG in the brain. And you can split it into harmonics. So each of these would be a component. So then you have an atlas of brain connectivity that covers whole brain based on functionality of the brain, not based on anatomy. And this is data driven. So you can pick one voxel and ask the question of which voxels are connected to it. So then in this way, you can actually identify different networks in the brain. For example, dorsal attention, ventral attention, language, or frontoparietal. So when I started looking at resting state functional connectivity of language and trying to figure out how it works for primary progressive aphasia, what was a hot topic at the time was this paper that came out from Marcel Meselem's team, showing that for people with primary progressive aphasia who have thinning of the cortex or atrophy in the left and only in the left temporal lobe, temporal pole, they have difficulty with verbal comprehension, with word comprehension. And so this is very lateralized. There's controversy and discussion in the literature as, it is semantic type of primary progressive aphasia. Is it a bilateral temporal or unilateral temporal involvement? Is verbal processing on one side or the other? But this showed that these individuals have problem with verbal processing, but not with visual and object recognition. So then there was a question of, is temporal pole part of the language network? So we approached that by looking at resting state scans we had for healthy or neurotypical individuals. And this was a study inspired by Liu et al. From the Randy Buckner group at MGH. They approached it this way. So if we put equidistance nodes and cover the perisylvian area, perisylvian language area, and its mirror side, its homologous right side, and look at all the connections between these areas, we wanted to see which areas are connected stronger to the rest. Liu et al. Did that. They showed that actually there is a big asymmetry between Broca's area and posterior middle temporal gyrus. So we did the same thing. We replicated that. So you see the posterior MTG and Broca's, and then there's also angular gyrus, and there is also temporal pole. And this has been shown in many other, and it's been supported in many other studies as well. So these 4 nodes to us sounded like they were very important. And so this was shown by Liu et al., the Broca's area or IFG, and then the MTG nodes. And then when we looked at these areas and the connectivity with temporal pole on the left side, we saw an asymmetric connectivity with temporal pole with both MTG and IFG. So that was very interesting finding, suggesting that temporal pole should be part of language network. To look at it with a data-driven approach, you'll see with ICA, again, you see a huge component on the left side, which overlaps with language network, and then you see connectivity with the temporal pole on the left side. So we use this model to see what's going on in primary progressive aphasia. And then I wanted to talk to you a little bit about how language is evaluated in primary progressive aphasia, so that when I talk about the different tests, it's not confusing. So usually with stroke aphasia or any type of aphasia, we do a naming battery, we do spontaneous speech evaluation, very commonly with the cookie theft picture, everybody knows that. They describe the picture, they name the items, and then comprehension by asking the person whether they can understand any of these sentences, and also repetition, which shows the connectivity between different posterior and anterior parts of the brain. In PPA specifically, we do a Boston naming test, of course. We use the WAP, picture description, Cinderella story. For comprehension, sentence comprehension, we use the Western Aphasia Battery and the Peabody Picture Verbal Test for comprehension of words. So they, individuals match words with pictures, with different pictures. They need to match them. And then we use the repetition subtest of WAB. For syntax evaluation, we use the Northwestern Assessment of Verbs and Sentences, NAVS. This is one example, for example, sentence production priming test, where we tell the person the dog is chasing the cat, and then they're supposed to say what's going on here. The cat is chasing the dog. Because there may be some verbal working memory difficulties in these individuals, our team came up with Northwestern anagram test, where it takes out the working memory component of it. So they are given these pieces of words on little pieces, and then they're supposed to make sentences based on the picture they see here. So the girl is tickling the boy. So I just want to give you some examples of how an individual with PPA would sound like. So this would be the cookie theft picture. Usually individuals can produce about 13 to 24 different components, and with a phrase length of average 4 to 5 words. And this is how a neurotypical person would say: a mother and 2 kids in the kitchen, the mother is washing dishes, the water is running in the sink. So let's see what happens with different subtypes of PPA. PPA is neurodegenerative disease. It gets worse, and then later it affects other areas of cognition too, but language remains to be the most severely affected area of cognition. It's usually caused by Alzheimer's disease or frontotemporal lobar degeneration. And different pathologies tend to be associated with certain subtypes. For example, frontotemporal lobar degeneration is mostly associated with agrammatic type PPA. So let's see if I can play this for you.
Can you tell me what's happening in the picture?
Obviously, man, a boy, and a cookie is on them, and his daughter, a sister, and when she's going to throw in with them, and they've broken a book on the store, on around one of them, a lady, and she was having a washing on them, and there'd be taps on them.
So this individual, as you see that the sentences are not quite grammatical, and then she does have anomia as well. As you see, she uses a lot of there and this and that and pauses. So, but you definitely see there's difficulty with grammatical sentences. This one is the logopenic variant, where people have more difficulty with word retrieval. They have phonemic paraphasias, meaning they make errors like cookie, the person may say cricky, phonological errors, and they have difficulty with repetition because of where the atrophy is, and it is in the way of the archaeofasciculus. So,
when you're ready, what do you see happening in that picture?
I see a mum washing, drying some plates, crikey, with water running down. I see a young boy in the other part of the
so this person, as you see, there are a lot of long pauses for word retrieval, but grammatically things are not as bad. And in semantic type, you have even more severe naming problem, object naming problem. And if you stop the person and ask them, show me the girl, they don't even understand what a girl is. It sounds to them like a foreign language. That's something that you can elicit in the cookie theft.
What's going on in this picture here?
Well, there was a woman and her children, and it's a house, I suppose, and there's a little window from there, and I don't quite know what that is going down precisely, and then the children are coonie jar. I don't understand that quite either, what they're doing. And gosh, that one's foot is about to come off. Oh dear.
It's very good. And what's happening over this side of the picture? Is anything going on out here?
Well, there's a people all down the end, I suppose. Just that way, or maybe not. Maybe it's buildings, maybe it's trees, etc. I don't know.
I think one hint was that the cookie jar was read as coonie jar, and he was not able to actually understand what it meant. All right, so the logopenic type is mostly caused by Alzheimer's disease, and the semantic type is mostly caused by frontotemporal lobar degeneration with TDP proteinopathy. So I'm going to present some of our research on what happens in PPA in terms of language network and functional connectivity. And from there, we'll discuss how we can actually help them. Our first question was really the relationship between function and structure. Well, we knew a lot about the atrophy patterns and maybe some correlations with atrophy and symptoms, but we didn't quite know where functional changes stood there. We did have a cohort of patients who had symptoms but had absolutely no atrophy. So those were a very good group of individuals to look at to see what's going on in terms of function. And then we wanted to look at more individuals we had with atrophy to see what the relationship between atrophy and resting connectivity is, how the pattern of these perturbations are different in subtypes of PPA, how do they connect or correlate with symptoms, and how can we use it for evaluation of individuals with aphasia. So to answer the first question, and we had 10 people with PPA who did not show any atrophy at FDR 0.05 compared to 35 control individuals or healthy individuals. And specifically because we wanted to look at, and these were all agrammatic, interestingly. And we wanted to look at the connectivity between IFG and MTG. We didn't pick the temporal pole because we knew that temporal pole is not involved in grammar processing. And then for control, we looked at a default network and prefrontal cortex and posterior cingulate cortex. We made sure that even at that level, there is no evidence of atrophy. There was no significant atrophy in any of those regions compared to the control group. And when we look at the connectivity between IFG and MTG, in that case, we showed that in PPA, the connectivity is reduced. And that was not the case with default networks. It was specific to the language network. And when we asked the same question using, and this was the node-based pairwise analysis with the seed-based whole brain analysis, you see that the connectivity between IFG here, where the Broca's area is, with the rest of the language network is significantly decreased. And this is in the absence of atrophy, because we didn't have to worry that atrophy is causing it, really. So this was a very interesting finding, and telling us that, you know, in certain individuals, actually it's the function that's abnormal and can possibly be modulated. So what happens in individuals with atrophy? And then we had 73 of those individuals, 36 in the agrammatic group, 20 in logopenic, and 17 in semantic group. You know, you see in the agrammatics, you see atrophy in the Broca's area. They all have perisylvian atrophy. In the real world, it's not that agrammatics don't have temporal atrophy. And then you see in PPA, you have more superior temporal, and then in semantics, you have anterior temporal atrophy. And they're all right-handed. You see the atrophy on the left side. We use the same pairwise model with 3 nodes. We did not use the angular gyrus because these 3 nodes were the most asymmetric. The angular gyrus nodes were not asymmetric, so we did not include in the beginning. We did later, though. And then we show that all PPA individuals had decreased connectivity between IFG and MTG. That shows you what an important connection that is for language processing. But the reason, of course, is different. You know, PPA-G has more problem with frontal lobe, AL has more problem with MTG, and semantics the same thing. In semantics, you additionally see the ventral network decrease. So this is compared to 33 control individuals. Then we try to factor in atrophy. Are these decreased connectivity findings because of atrophy, or are they beyond atrophy? So we found that in semantic and logopenic individuals, they had proportional atrophy. So when we corrected for amount of atrophy, there was no significant finding. But in agrammatics, the decreased connectivity was above and beyond the level of atrophy. So that tells us maybe in agrammatics there is more room for neuromodulation because it's more on based on functional changes. And this actually followed the same pattern as the other group that we looked at without atrophy. They were agrammatic too. They had more functional rather than structural changes. Well, so other people had looked at these in PPA. We were not the first one to look at these disconnections in PPA. What was not really very clear in the literature was how do these connectivities correlate with symptom. So we do a lot of testing in our center, and we were blessed to have a lot of information about these patients. So for repetition, we found out that this IFG-MTG connection correlated with repetition. It also correlated with grammar processing. And when we looked at naming, with Boston naming and word comprehension with PPVT, we found out that it's the ventral pathway, the MTG connectivity with ATL, anterior temporal lobe, that was decreased. So that tells us two things. First of all, the MTG is such an important node for both lexical, semantics, and grammar processing. And it's this connection with IFG is more associated with production, repetition, and grammar, and with ATL with lexical semantic processing. So the findings, as you've noticed, are not really specific for agrammatics and logopenics. We wanted to find a way to be able to distinguish them based on their pattern of language disturbance, language network disturbances. When we looked at seed-based analysis, we found some answers there. So as you see for the IFG or the Broca seed, there is more decreased connectivity with the premotor areas of the brain. And that's why you see in a lot of agrammatics later, they can actually have trouble with motor speech also. They can have progressive apraxial speech, but logopenics usually don't. And then, so that can distinguish the two. And this is the language component that you saw earlier. And then in semantics, there's really nothing in the dorsal pathway. So this was interesting. The connectivity, first of all, as you see in the ICA and here in the PPA, the connectivity between the perisylvian areas and dorsal frontal areas is very interesting. That is actually the bridge between linguistic and social communication, the link between perisylvian language and then the frontal lobe. And so same thing we did with temporal pole or anterior temporal lobe. I looked at the premotor and superior frontal gyrus areas, which are the peak areas of connectivity. If we put a seed in IFG. And then if we look at the connectivity on both sides, we found that, again, there's asymmetry between connectivity of dorsal frontal areas and language areas. So this has implications, for example, for neuromodulation. If we can't get these nodes here, can we use the dorsal areas to get to them? And then we specifically looked at this connection, the superior frontal gyrus and Broca's area or IFG, the triangularis part. And we found out that in agrammatics, this connection is indeed decreased. And in logopenics, instead, you see the connectivity between angular gyrus and IFG is decreased. So we really added more locations now, more regions of interest based on the literature. We did include the posterior STG2, but didn't find much with posterior STG. That's why, you know, when in the functional literature and with the structural lesion-based literature, you see there might be a little bit difference in terms of where Wernicke's area is located. It's definitely in the posterior part of temporal lobe. And in terms of repetition, as we know, it's related to the temporoparietal junction atrophy in logopenic patients. And Marco Catani has also looked at it with tractography. There's this interesting tract that goes from Broca's area to the superior temporal frontal lobe. And it's called the Aslan tract. So that makes sense anatomically too, this connection. So when we looked at the regions of interest, pairwise analyses, and we looked at right hemisphere, we really didn't find any change in terms of decreased connectivity. Right hemisphere always remained comparable to controls. What we never did actually was we never looked at whether right hemisphere had actually increased connectivity. And then when we asked that question by putting the seed on the homologous areas or the mirror areas on the right side, we found that actually in each of these subtypes, there are areas that showed increased connectivity with the seed that we are using. The one that really impressed us was in logopenic individuals, where it actually, if we put a seed on the right side in the anterior temporal lobe, you see increased connectivity locally and also with the left temporal pole. And this connectivity, if we look at the correlation between the connectivity values and Boston naming test, we found that it correlates. So these are individuals who have atrophy here, but they don't have much atrophy in the temporal pole. So this seems to be a compensatory mechanism, which again has implications for interventions. So I want to talk to you a little bit about how we can use functional imaging for assessment of treatment. So this is a case study we did when I was at the University of Arizona. It was for a logopenic PPA patient who went through semantic-based intervention, where the individual had to describe the object, and then through that would actually access the word. And we showed that after the intervention, there was more activation in the dorsal frontal area, which is actually connected to the semantic system. We did another study with Dr. Maya Henry's lab in University of Texas, Austin. She's also doing interventions for primary progressive aphasia. She uses a method called video implemented script training, where they learn how to make patients learn how to produce sentences based on scripts, and then they do this with video implemented administration. They can do it at home as well. So for this one, because we had only 5 patients, we didn't have enough power to use connectivity analysis. We used amplitude of low frequency fluctuation, the power of resting state, which was decreased, of course, blue in these areas. And after treatment, we showed that there are areas that are increased in terms of that amplitude. So that can actually tell us the changes. And these are people who did respond to treatment, both sides. We can actually see the neurocorrelates of that responsiveness to treatment. And so these can also be used, when we know more about the network, we can use it for neuromodulation. And I know your center has a very great track record in neuromodulation, and I don't need to talk much about it. So with transcranial magnetic stimulation, we can stimulate or inhibit areas of the brain. And I am collaborating with Joe Voss's lab at Northwestern, who's done a lot of work with memory. And he showed that actually areas of the brain that we can't get to, for example, temporal pole is very difficult to get to, as you know, because it's very painful to stimulate over the temporalis muscle. What can we stimulate other areas of the brain and get to, for example, some area like hippocampus, which is deep in the brain. And Joel has shown that, yes, that is possible. So we are trying to do the same thing to see if we can get to those areas and do some neurostimulation or inhibition. So let me summarize. Thank you for bearing with me. I talked a little bit about our evolving understanding of language network and how we started to learn from stroke patients, and later with newer technology, with tractography, functional imaging, better structure imaging, we know better about the language network. We learned a lot from primary progressive aphasia because it affects the brain differently. It affects the cortical areas more than white matter compared to stroke. And the big idea right now is that the organization within the dorsal and ventral pathways. In PPA, we found that the connectivity is impaired even before there is evidence of atrophy. So resting state is a leading indicator for the disease. And word comprehension depends more on the ventral nodes that we looked at and syntax on the dorsal ones. And abnormal word comprehension in PPA semantics seems to be proportionate to the amount of atrophy. In agrammatics, we saw that there was more role for the functional disturbance rather than structural. Yes, you already said that. And a better understanding of functional derangement with the language network can help us with measurement of responsiveness to treatment. And even prediction on a case-based analysis, if we finally can do machine learning approach based on the pattern of atrophy, the pattern of functional connectivity. If we include PET scan, which I didn't have time today to talk to you about PET scan, we've been looking at the fluorodeoxyglucose scans as well. Can we say who's going to respond to treatment? And I know, again, your team has looked at these questions in stroke patients beautifully. And these are the superstars in my lab. Some of them still working with me, some of them graduated. Without them, I couldn't have done all that I showed you. And I want to thank people in our center, Dr. Meslem, Robert Hurley, who really, we worked together to figure out the resting state in healthy individuals in PPA, Emily Rogalski, and many others in our center. Todd Parrish in Center for Translational Imaging, and many collaborators in different universities, Stanford, Mike Gricius, Harvard, Randy Buckner, and our funding sources. Thank you so much. I am here for any questions you might have.
Thank you so much, Borna. You only see me clapping, but obviously at home, everybody is going wild now with applause. So I invite the audience to type their questions in the chat box. Just to get us started, I was wondering, when you discussed your 2019 paper, it seems almost as if you discuss functional impairments and structural impairments as dichotomous. And I'm not sure that's actually what you mean, but so it seems
not really. You're right. No, right. They're not. They affect each other. So it's not that they're separate from each other.
Well, I would say any functional deficit has a structural basis, right? Even if you're not able to measure it. So in that sense, I was wondering whether the prediction that you made that the patients who only show, as you say, a functional deficit without atrophy being significant, whether that really is a dichotomous.
Right, that's a very good point, Derek. You're right. So it's not that these individuals don't have atrophy. We have a threshold we look at, and then, as I said, it's the FDR05. I mean, if you change the threshold, you may see something, bleeding through. But at that threshold, if they had atrophy, it was really minimal.
Mm-hmm.
So you're right. Yes, it's not that they do have pathology, of course, and then they do have cell damage. I think at that level, we didn't see. So these were really the mildest patients we looked at who didn't have as much change in structure. But yes, that is always, it's a tricky business when you say there's no atrophy. Yeah, according to what threshold, you're right.
Yeah. And of course, there may be other, what I would still call structural damage.
Oh, absolutely. So for example, what we haven't looked at is the tractography. We don't know what's going on in their DTI. Like if we do tractography, maybe they have changes there. Those are structural changes we haven't looked at. The other thing we haven't looked at is basal ganglia. We know that in the frontotemporal lobar degenerative patients, like PPA, agrammatic, or semantic, they do have abnormalities in basal ganglia. We have started looking at these. We have, in the studies I showed you today, we hadn't looked. So absolutely, it's not true that there's absolutely no structural change. But in neurodegenerative disease, what happens, the series of events is usually you have these abnormal proteins accumulating. Then first they disrupt the function and then the structure. So there is, however, again, you're right. It's not, you can't, so based on the 2011 Clifford-Jack model, it was first function, then structure, but then later they said, well, it could happen at the same time. So it is tricky.
Thanks. Questions are flying in now. So here's a question from Julius Fredrickson. Thank you, Borna, for a wonderful talk. I have a question regarding what is known about the relationship between gray and white matter atrophy in PPA. Is it possible to distinguish between PPA types solely based on white matter atrophy?
Great questions. Hi, Julius. So Julius, like Dirk, is my aphasiology brother. We were trained by the same people.
So that's a great question. We haven't actually looked much into tractography, very limited at our center. But my colleagues at UCSF, they've looked at the tractographic changes in PPA. Usually in semantic patients, they do have more ventral tracts affected, for example, the occipitotemporal or the external capsule. And in agrammatics and logopenics, you have more dorsal pathway. And then in logopenics, you have more posterior dorsal pathway or arcuate, and in agrammatics, the anterior. We usually don't use imaging data to subtype PPA patients. We do it based on clinical findings. Other centers may actually include that for subtyping. We use it only for clinical purposes because there's overlap. Some people with agrammatism may actually have the pattern you see in logopenics. They have posterior atrophy. But yes, looking at white matter, which is what I haven't specifically looked so far, that can definitely add to better understanding of what's going on.
Thank you. So I think, so we have a question from Maria Ivanova, which I think is related to both of the above questions, but maybe you can expand where necessary.
Sure.
So thank you for a great talk. My question is somewhat related to Dirk's question above. What do you think underlies the observed functional impairments in PPA? Is it changes in gray matter that we are not yet able to detect with our current neuroimaging analysis? Or could it be deterioration of white matter networks that support the explored functional networks?
Hi, Maria. That's a great question. I think it's both. We haven't looked at how white matter really contributes to these changes. It's a very big one because in FTLDs, there is white matter abnormalities. So part of it is definitely, I can imagine white matter changes, but we still need to look into it. And there's also synaptic failure that we see in neurodegenerative disease before there is cell death. So what's happening is because of these inclusions in the cells, they are not able to communicate with each other. So this is really a gray matter phenomenon. But at the same time, there are changes within the axons, within the tracts, which affect the connectivity of areas. So yes, the answer is both. Both can contribute.
So we have a question from Bree Stark. Your regions of interest were anatomically defined. Do you think we could account for more variance if we stay at the single subject level, that is, parcels fit to each subject-specific brain, rather than molding brains to a shared template? Would looking at strength of connection between resting state-derived networks, for example, cognitive control versus language, also be a viable next step in PPA?
Okay, let me, this was beyond my working memory. Let me answer the first question. So the first question is, yes, we can't really be rigid in terms of what areas we are choosing. This was just at group level analysis. Of course, at single subject level, we have to be flexible. And in those cases, actually a data-driven approach would actually be much better. So if we want to really look at single subjects, it would be better to use, for example, ICA, which is what we've actually started to work on. And then what's the second part of the question?
So the second part was, would looking at strength of connection between different resting state derived networks, so for example, between a cognitive control network and a language network, also be a viable next step in PPA?
Absolutely. And then we have started to look into that. For example, frontal network and language network, they work very closely, as you saw in the language component of the ICA. It goes all the way up in the frontal lobe. And we do see, for example, one of the things that we are looking at is patients with frontotemporal dementia, if they have executive dysfunction, they do have word finding difficulties. And then we are trying to look to see how much of it is language network and how much of it is actually the frontal network. If you ask them to come up with names of animals, they can't come up with more than 4 or 5 sometimes, although they are not overtly aphasic. So yes, looking at the interaction with other networks is definitely the next step.
Okay. Then we have a comment. RG Hillis's iPhone says, great talk, thank you. And Ajay Kurani says, great talk. Then we have a
Oh,
question.
great.
From,
Hi, RIJ.
we have a question from William Matchin. Have you thought of defining your analyses using functional impairments, different types of grammatical, semantic, and lexical deficits, for example, rather than clinical diagnoses?
Yes, so that is being done in our center. For this purpose, actually, that's a very good comment, because technically we can use those kind of pieces of information for our studies as well. For our interventions, we have a group, they are doing telehealth or teletherapy, because our PPA patients are all around the country. They do specifically look at functional and quality of life measures. And that's a very good point. Actually, we can look into those too, because that's where really what matters in terms of real life situations.
Just to add to that, I think William also meant, rather than by clinical diagnosis, going by language component. So grammatical versus semantic versus lexical, and to categorize patients like that rather than categorizing them.
Oh, right. So that's another good point, that yes, we don't have to actually come up with the subtypes, because in real world, individuals can actually go across subtypes. So you can have someone like the first patient that I played for you who had agrammatism and did have also anomia too. But in pure agrammatism, you usually don't have as much anomia. So yes, you could go by, and especially for interventions, for speech pathological interventions, you go by the functional impairment rather than, treating logopenic. So no, there is no prescription to treat logopenic PPA, but rather treat anomia or treat agrammatism based on their functional impairment. Very true.
And a question from Brian Chiappetta. Great talk. You might have said this, but can resting state connectivity predict the progression of atrophy in people with PPA, meaning how quickly different regions atrophy over time?
Yes, definitely. And then our colleagues at UCSF have done extensive research on this. They, Bill Seeley and his group, and later many others at UCSF have looked at the fact that when you have these resting state networks, you see that the atrophy patterns actually follow these networks. And they actually had a recent paper that came out a few months ago that they do predict what will happen if you have connectivity disturbance in areas of the brain, what happens in terms of atrophy. So yes, you can predict that. That is possible, and people have done that.
Okay, we have a question from Elena Barbieri. Thanks for the great talk, Borna. In the case of agrammatic PPA, where you found that resting state connectivity disturbances are not accounted for by atrophy, can you comment on if those participants had subcortical atrophy? We followed an individual with agrammatic PPA for 2 years, during which he only showed subcortical, caudate, and hippocampus atrophy. So I'm interested in getting your input on that.
Right, that's I think in line with other questions, that yes, in these individuals who don't have as much cortical atrophy, they are, and I think your patient, Elena, definitely shows us what's going on in these individuals. Yes, they do have some subcortical changes that we haven't looked at. So as I said earlier, yes, you can have the changes in your tracts, you can have changes in basal ganglia, and that can be the reason for their symptoms, on top of actually cortical connectivity itself. So it becomes a very complex situation where there's cortical dysfunction and also connectivity with deeper structures, and also the highways connecting them.
Thank you. And the last question is from Kirana Tsapkini. Great talk, Borna. Could increased connectivity also be a compensatory mechanism showing effort?
Showing effort?
So in some cases, you might see increased connectivity. And the question is always if that's the impairment or if that's the response to the impairment, right? I think that's how I would interpret Kirana's question.
Oh, I see. Well, I mean, very true. So in the logopenic patients, it was really, it looks like, because it correlates with their better naming. So I would want to say it's compensatory. In the other subgroups, we are really trying to figure out, it doesn't completely correlate with symptoms, which means it could be just effort and it doesn't really go anywhere in terms of compensation.
It's an attempt at compensation.
Yeah, right.
Okay. And then I have a comment from Ayana Tiptop. Thank you for a great presentation. And
Thank you for very stimulating questions.
thanks a lot, Borna.
Thank you.
So next week, everybody will be listening to Amy Dietz. That's June 18th. And for now, we thank Dr. Bonakdarpour for his presentation, and hope to see everybody at one of our following C-Star talks. Goodbye, everybody.
Thank you for having me.